I'm the Chief Medical Officer of Mirati Therapeutics, a biotechnology company located in San Diego. And we are primarily focusing in drug development for oncology, for cancer treatment. And our philosophy is that everything that we do has to be based on genomic selection of the patients who we believe is going to respond the best to our drugs. So that's the premise of our company. So we have 2 kinase inhibitors and 1 epigenomic compound, HDAC1, 2, and 3 inhibitor. And with every clinical trial, we have a diagnostic in mind. So our development and the premise of this strategy is that we can benefit the patient the most with a relatively small clinical trial study, and that can lead to hopefully to the approval of the drug in a relatively small number of patients in a very efficient way. Personalized medicine is having an incredible impact in the way how we think about drug development and also clinical trials. It's blurring essentially the traditional stage of phase 1, phase 2, and phase 3 studies. Early on, even before we test the drug in patient, we are already thinking what's the strategy. And you still need to go through the safety and dose escalation to find the ideal dose for the patients. But the traditional concept with chemotherapy, such as reaching the maximum tolerated dose, which is essentially try to test what's the amount of drug that you can give to a patient so that they can tolerate, they don't get excessively sick. I think that concept is changing already to what's the optimal biological dose instead of what's the maximum tolerated dose. So that's one first concept. And the second concept is that early on, right after you find out what the recommended phase 2 dose, and I'm not going to mention maximum tolerated dose, which it's what we are trying to move away from. Once you have a recommended phase 2 dose, what's the patient population you want to target? Which patient are going to benefit the most? So that even in the early phase 1 study, you can have a proof of concept that the genomic alterations that you're looking for are the ones that predict a response and maximal benefit from your drug. So that's what we're doing. In a phase 1b expansion, we already have a diagnostic test for those patients, and we are selecting patients with tumors that we believe are the drivers, and those patients will benefit the most from our treatment now. Another change in the concept in the past is to treat lung cancer, bladder cancer, divided by disease type. We have a basket early on in the dose expansion, what we call a basket group, which is tissue agnostic. We don't distinguish whether the patient has lung cancer, bladder cancer, gastric cancer. If the tumor have the genomic alterations that we're looking for, they are eligible for the study. And the premise of this is that if you truly believe that those are the driver mutation, it should not matter what kind of tumor they have. And that's the genomic alteration that is driving the progression of the disease. And therefore, your drug should work across the board in different tumor types. So those are also a different concept from the traditional clinical studies. Of course, that has a lot of regulatory implications, but it's very exciting. I think people are changing the concept. The FDA, it's open to this kind of innovative thinking. And I think that you will see more and more based from the recent announcement of recent statements from Richard Pazdur, Dr. Pazdur is the head of Division of Oncology for drug approval at the FDA, that they are very open to this concept, and they're also catching up with the technology as well. The FDA has acknowledged that the science is progressing very fast, and things that we didn't think about or cannot even think that was possible 5-10 years ago, it's happening now, especially with molecular testings. Genomic screening in the past used to be very expensive. It's still expensive, but the costs are coming down. So it's feasible, it's practical now. And the technology is changing so much now with next generation sequencing, for example, in which you can test several genes, alteration in the genes at the same time. Instead of sequencing, testing one gene at a time, you can test with one tissue sample, you can test several genes alterations simultaneously. So the FDA acknowledged that now. And also, technology is changing now because tissue is very hard to obtain depending on the disease, such as lung cancer. Tissue are very precious, and sometimes you cannot just go back and biopsy the patient over and over again. And now the concept of liquid biopsy or circulating tumor DNA has the potential to change the landscape significantly. And the FDA acknowledged that. And I think they acknowledge also that the trial design that drug companies are doing is changed from the traditional phase 1, 2, and 3. Like I alluded to previously, they acknowledge that a lot of drug has been approved sometimes even with dose expansion from the phase 1 study. And that's because if you are right, if the science is correct, you see a huge benefit that was unthinkable with traditional chemotherapies. Now you're benefiting patients that even in a small number of patients, you can see such a treatment effect, magnitude of treatment effect, that it's undeniable. It's hard to later on to think about thousands of patient study when you have 100, 120 patients and have response rates in the 50, 60% rate. And not only response rate, but very good quality, very durable responses. So FDA has been on board. They're trying to help with this breakthrough designation. I think it's very helpful to companies like mine, a relatively small biotech company. And that's not to say that there is no challenges. When you talk about personalized medicine, you're talking sometimes screening 5% of the patients. What that means is that you have to screen 20 patients to find one patient that's eligible for your study. But that one patient benefits significantly from your treatment. So that's a trade-off. And one of the challenges is to keep the investigators motivated because they have to test many patients to find one that is eligible. So there are challenges to it. And ironically now, the significant portion of the cost of the clinical development, it's not that much in the patients. In the past, we used to have thousands of patients. Now you can get your drug approved, if you're right, with 120, 140 patients. But half of the cost can be in the genomic testing now. So there is a shifting, it's a trade-off. But I think the payout is significant for patients' benefit and also for drug companies as well. Some of the key challenges in personalized medicine is to find out what's the feasibility of the target gene that you're looking for. Just an example, MET, for example, is an oncogene. And if you are looking for a mutation, activation mutation in the MET, for example, what is the true incidence of the mutation in lung cancer patients, for example? And depending on the data that you assess, the incidence or prevalence can vary from 1% to up to 8%. But what is the true prevalence of these mutations? So what we have been doing is to search for available data, such as the Cancer Genome Atlas, or even collaborating with companies such as Foundation Medicine to try to figure out what's the true prevalence of the mutation. And that has practicality issues, whether you can find, if it's less than 1%, it's going to be very difficult for you to run a clinical study. But if it's 5% or more, then it's viable. And so that's one challenge. What is the true prevalence of the mutation that you're looking for? And which database can you do your confidence that if you're going to pursue that route, and that's going to be successful? The second key challenge that I think is the technology. There are so many ways to find the mutations now. There's next generation sequencing that you can test several genes simultaneously, but then the challenge of that is what is the mutation that is clinically meaningful? Because the test is so sensitive and powerful, you're going to see mutations that might not affect the function of the gene, and which ones are really relevant for the patient and to change the biology of the tumor. And there are other technologies now. Do you use next generation sequencing, which is more expensive, or do you do FISH? Do you do RT-PCR? And now tissue is very hard to obtain. A lot of times they are very precious. You cannot just go back to a patient and keep on biopsying, such as lung cancer. It's restricted, the amount of tissue that you are able to obtain. So now the concept of liquid biopsy of a circulating tumor DNA, for example, that's very attractive. It opens up to potentially ability to screen more patients. So those are the kind of technology that you have to decide before you even start a potentially registration study, which one you're going to use for your patient. And that's a dialogue that you have to have with the FDA to make sure that everybody's on board before you embark on a large clinical study.